Hey students, this is Mrs. Foy, and this is a screencast on genomes and their evolution. I think one of the most interesting things that I have read recently compares what is going on in biology right now, in the past 25 years, to the golden age of exploration in the 15th century, where, as you know, in the 1400s, there was all this exploration of the world because humans designed better ships and because we had better navigating systems. And the explosion of what we have learned about after sequencing, just first of all, finding the structure of DNA and then being able to sequence DNA has ushered in this golden age of biology that you're living in right now and studying AP biology right now. So that's pretty exciting. So we know from the lab that you guys have done with the blast and identifying the bacteria that there are huge websites powered by supercomputers that will allow scientists all over the world to compare gene sequences and also protein sequences. We know that scientists are not only studying genomics, but they're also studying proteomics, which are looking at the structure of proteins and comparing them to other organisms. And from this, we can tell evolutionary relationships. And because of this, we actually have a whole new area of living things that we call the archaebacteria. We know that they are so different in their protein structure from the other bacteria that we have that we had to rearrange the whole tree of life for them. So we know that when we look at the DNA and be able to look at what that DNA codes for, we can find some very, very powerful ideas in biology. And one of the things that we know is that we know that a lot of the genes are conservative. That means they stay in place as we go through the evolutionary tree, which of course is amazing since we have such a diversity of life. But the fact that we do have sequences of DNA and also proteins that those code for that are similar in organisms in this tree of life allows us to find evolutionary relationships, just like a family tree that you might search. So remember that only about 1.5% of the DNA codes for proteins and or RNA, specific types of RNA like transfer RNA and messenger RNA. And we know that most of the DNA is non-coding DNA. And we used to call this junk DNA, but now we're finding that the non-coding DNA is also highly conservative, which means that there's a lot of evidence that the fact that it hasn't changed much must mean that it is serving an important function, and we're just now starting to find out what those functions are. Both eukaryotic and prokaryotic cells have stretches of DNA that can move from one section to another. And this discovery was from a really cool scientist named Barbara McClintock. And Barbara McClintock worked on corn. And she actually was looking at certain types of maize that she was actually looking at the pigmentation of the different kernels in corn. And she discovered in the 1940s and 1950s that these genes had movable chunks called transposable elements. And interestingly, her work was laughed at and not accepted for a long time. And then scientists, which often happens with somebody who's revolutionary, began to recognize and have other people be able to back up her data. And she eventually won the Nobel Prize in 1983 at the age of 81. But what Barbara McClintock found is that some of the non-coding DNA is a type of, can undergo a type of recombination called a transposition. And these transposons, so these sections of genes, can either be a cut and paste or a cut and copy type. So you can have a section of a gene, this transposon, that can either cut out and then repaste in another section, or it can be copied and pasted like you see in this diagram. And interestingly, about 25 to 50% of the non-coding DNA in mammals is the transposable type, and as much as 85% in plants and amphibians of the non-coding DNA is this transposable type. So it's very, very interesting. Another type of transposition is caused by a little bit different type of transposon called a retrotransposon. And what happens with these, and we're not really sure why this happens, but a piece of messenger RNA can be, instead of going on then to be translated, it can be acted on by a reverse transcriptase and then get plugged back in to the DNA itself. So that's called a retrotransposon. And of course, both of these transposon types are going to increase variety. This is another source of variety in the genome. One other interesting point about, I'll go back just for a second, about some of this non-coding DNA. We know that it is repetitive in nature, and interestingly that some of this repetitive DNA is found in certain places on chromosomes, like right around the centromere region and also on the ends and the telomere regions. So that is an interesting point that we're going to come back to later. So some of this junk DNA, so to speak, are pseudogenes. And pseudogenes are genes that used to actually code for a protein, but they have undergone some type of mutation that has made them useless. But they are still persistent in the genetic code. And one of the ways that a pseudogene can become useless is because of, say, a retro transposition that could occur that would then mess the gene back up and not be able to code for a protein anymore, or sometimes duplications also can cause pseudogenes. So pseudogenes are interesting to trace because they're kind of dead ends of genes, but they give us information about evolution and how genes have changed. So one of the most interesting things about our closest relative to humans are chimps. We know that chimps and humans are very, very close genetically speaking, but one of the things that's different about us is that chimps have an N of 24 chromosomes, and humans have an N of 23. So as we began to learn more about chromosomes, we began to look at chromosome number 2 in humans. And chromosome number 2, as you know, is one of the larger chromosomes because in a karyotype it goes from the smallest number to the largest number. So, for example, chromosome 22 is the smallest chromosome. Anyway, a human chromosome number 2 is very interesting because its telomere sequences and its centromere sequences are very similar to 2 chimpanzee chromosomes, chromosome number 12 and chromosome number 13. And so scientists have known that somehow along the 6 million years that separate us, the 2 chromosomes in chimps, chromosome 12 and chromosome 13, actually fuse to form the human chromosome number 1. And interesting that a lot of the places on chromosomes where a lot of transposons and a lot of rearranging goes, we call those hot spots, places where the chromosomes can break easily. Those are actually, they have a correlation to places in the genome that are related to congenital disease. So that's very interesting. In this case, we can see an evolutionary pathway back to our ancestors. This is pictures of some very conservative genes in a mouse chromosome that are very similar to the chromosomes, the genes that we see in humans, except in mice, they're on different chromosomes. Here, mice chromosome 7, 8, 16, and 17. And in humans, we find them all on one chromosome. So again, looking at differences which help us to be able to find similarities in our evolutionary background. So we have learned about crossing over between homologous chromosomes, and we can see that sometimes genes duplicate due to unequal crossing over on non-homologous chromosomes. And we know that these transposable elements, these kind of mix and match sections of genes that can get out, can cause some uneven crossing over to occur that are going to result in new gene combinations. And these new gene combinations are going to be the source of mutations and new gene combinations that fuel evolution. So one of the genes that we know more of the evolution of, this is an example of how you can get a new gene by shuffling the exons of some other genes. So, for example, we have a gene for epidermal growth factor gene that has multiple exons for that gene. And then we have a fibronectin gene with multiple exons. And the TPA gene in humans exists today as a shuffling of the exons from those different genes. And suddenly we have a new gene. We have an evolution of a new gene that we can trace. By looking at the relationships of the DNA, scientists have determined that all life can be grouped into 3 different domains. So these are big groups of living things. And we know those 3 domains are the bacteria, or the eubacteria, the true bacteria, the eukaryotes, so these are going to be all organisms that are made of eukaryotic cells, and the archaebacteria, which are very ancient, kind of weird bacteria that live in very extreme environments. Sometimes we call them the extremophiles. So they would live in very hot areas like thermal springs, or very salty environments. Those are called the halophile bacteria. But we see the evolutionary pattern of these 3 domains. So about 3.5 billion years ago, we have the breaking of the bacteria branch and the eukaryote archaebacteria that then further branched about 3 million years ago. And then you can see about 6 million years ago, we had a common ancestor of chimps and humans, and way back in 65 million years ago, around the time of the dinosaurs, that is where there was the last common ancestor between humans and mice. But one of the genes that is very interesting today is this gene called the FOX gene. We know that there are certain genes in humans that are rapidly evolving. Because we can see how fast they're changing, because we can look at our ancestors' genes and we can see that they are changing very rapidly. And one of the very fast evolution in genes is our body's defense against malaria, our body's defense against tuberculosis. So this is kind of an arms race that we're undergoing with protists in the case of malaria, and bacteria that cause tuberculosis, but also in the genes that determine the size of our brains. And we can look at genes that code for transcription factors, because those are going to help to regulate the gene expression. And one of these genes that we see that scientists are doing a lot of experiments with is this FOXP2 gene that's found on chromosome 7 in humans. We know that this gene serves to help direct the speech production and language in the human brain, because when there is a mutation in this gene, then there is severe speech problems and language impairment in humans. We also know, this is so cool, that this FOXP2 gene is also the gene that gets turned on in certain songbirds like zebra finches and canaries. And we see that this gene turns on right during the development of when these birds start to learn their songs, their species songs that are significant to those species. So one of the things that scientists have done is they have been able to take the humanized copy of the FOXP2 gene and they have inserted it in mice. And one of the things that they saw is they saw that these mice had subtle changes in their vocalizations. So mice have vocalizations. They usually make these at a type of sound that humans cannot pick up. But they could see that there was a subtle difference in that, and also in the circuitry in the mouse brains that is the same as the speech areas in the human brains. So in 2005, scientists were able to knock out this gene, this FOXP2 gene in mice, and they were able to measure the number of whistles. So these ultrasonic whistles that mice make. And in the wild type, which did not have the mutation, they could detect with instruments so many whistles. In the heterozygous, so this would have one normal gene and one of the mutant genes, there was significantly less. But in the homozygous mice that had both copies of the FOXP2 gene knocked out, the mice were unable to make those sounds. So another area that is a really interesting area in biology is called evolutionary developmental biology. So evolutionary developmental biology, or evo-devo biology. If you Google this, there's tons of stuff that comes up, and it's very, very interesting. But one of the things that's really cool about developmental things in biology is that they are very similar. So they are very conservative. So for example, here you see the embryos of fish, reptiles, birds, and mammals. So, these are very different branches of the tree of life, but you can see very specific body plans. You can see gill slits that are in all four of these. You can see tails, you can see limb buds, you can see segmentation of what will become the muscles and bones of these animals. And so we know that that there is a conservation of the developmental genes in different animals. And one set of these genes are called the homeotic genes. So for example, in fruit flies, we know that there is a specific set of these homeotic genes, which is kind of like a master switch for developing the body segmentation of fruit flies. And what is so cool about this is that these genes are so similar to the basic master switch genes in our development that one scientist quipped that fruit flies were actually little people with wings. They are so similar. So we call these sets of genes Hox genes because they are homeotic genes in a homeotic box, which includes some certain segments of bases that also include the regulatory sections. So if you look here at the Hoxbox genes in fruit flies compared to the Hoxbox genes in a mouse, the colored bands that you see are the genes that are the same. The black areas are the genes that are different. And so you can see one of the things that's different is that these Hoxbox genes are on one chromosome in a fruit fly. Fruit flies only have 4 chromosomes, and they are found in different genes in a mouse. But the comparison between these genes is amazing. So you have anterior and posterior sections, and then this body segmentation that would then develop as kind of the master body plan of both of these two animals is basically the same. So One of the things that is a burning question is that how can the same genes be involved in these two organisms when the animals themselves are so different? And that's one of the burning questions of biology that is yet to be understood. Right, just very quickly before we finish up, I want to talk just a little bit about animal and plant development. So, when you look at animal and plant development, we diverged a long time ago. So, the last time we had a common ancestor was, sometime a little before 2 billion years ago. About 2 billion years ago was the first time we had eukaryotic organisms, and the first plant cell we began to see not so long after that. So we diverged a long time ago from each other, but even though plants and animals have very different body structures, so plants develop these very rigid cell walls, and animals have more movable parts, movable cells that migrate and movable tissues that migrate, and the rigid cell walls in plants prohibit that. But scientists have discovered these master regulatory switches in plants called MADS-box genes, and they seem to have a similar type function as the Hox genes do in animals. So even though some plants have Hox genes and some animals have the MADS-box genes, they seem to have developed independently of each other. And so this is just a little introduction about how genes evolve, and I thought it would be a basis of understanding for you as we begin to explore a little bit more about evolution. So I hope you've enjoyed this and hope you learned a little bit.